chart that should help us remember them. And we're going to be looking at some genetic characteristics that are found in people, and we're just going to look at yourself as an example. So you probably want to have something, you know, some sort of mirror handy that you can look at some of these characteristics to be sure what your answer is. So first of all, let's take a look at the chart. We have a chart and we're going to look at 9 characteristics that are all genetically determined in people. These are characteristics where basically there are 2 possible alleles. So you could think of this as 2 factors, like Mendel's factor for yellow peas versus green peas, or round peas versus wrinkled peas. And then we're going to look at the phenotype that we can observe. And remember, we said when you see phenotype, think pH phenotype, pH physical characteristics. We're going to look at the genotype that goes with that. And we're going to look at, you know, if you have that genotype, what type of gametes, what type of eggs or sperm could you make? So let's start out by looking at tongue rolling. And tongue rolling, what we're looking at is whether you can roll your tongue into a U shape. If you can't roll your tongue just kind of flops back and forth and nothing happens. And this is actually genetically determined, so you can't do a tongue workout and somehow become able to roll your tongue. So there's 2 possible phenotypes. Now remember, the phenotype is your physical description. So let's change this. We want to have our 2 phenotypes on different lines. So either you can roll your tongue or you can't roll your tongue. Now I'm writing both of these down, but you're just going to write down whatever your characteristics are. So if you can roll your tongue, you're going to write that down. If you can't, you're going to write that down. Now, the ability to roll your tongue is caused by a dominant allele. So if you can roll your tongue, you're big R, big R. We use capital letters to designate dominant alleles. Or there's another possibility. Remember what Mendel said, one allele can hide the other allele. The allele that hides the other that we see is the dominant allele. So you could be big R, little r. Now, if you're big R, you got a big R from both of your parents, from your mom and your dad. If you're big R, you got a big R from one of them, a little r from the other one, and it doesn't really matter who you got the big R from and who you got the little r from. If you can't roll your tongue, there's only one possible genotype. Your little r, you're homozygous recessive. Now, what about the gametes you can make? What about your eggs or your sperm? Well, if you're big R, all of your eggs or sperm will have big R in them. Keep in mind, you only pass down one R because the other parent passes down the other R. Or the other possibility is if you're big R, then half of your eggs or sperm get big R, half of your eggs or sperm get little r. Let's make our font smaller here. I want to keep everything on the same line if it's related. So if you can roll your tongue, you should have this entire first line written down. So your phenotype, you can roll your tongue. Your genotype, big R or little r. And then your gametes, either all big R, if you were big R, or half big R, half little r, if you were big R, little r. If you can't roll your tongue, then your genotype is little r. Or the other possibility is Whoops. All your gametes are little r. So if you can't roll your tongue, you should have this section that's highlighted in green written down. So you should have either that top line written down here or the bottom line. Now, what do you write down? Well, it depends on what your phenotype is. The phenotype is what we can look at and see. The genotype is what we can deduce from your phenotype. We've done some studies, so we know which allele is dominant here and which one is recessive. And the gametes come from the genotype. So are there any questions about how you would fill this first line out? Okay, for the second characteristic, we're looking at something called widow's peak. If you have widow's peak, your hair comes to a point in the middle of your forehead. If you don't have widow's peak, you have a straight hairline, which isn't really straight. It usually does curve up a little bit towards the top, but you don't have that point. Now, some people shave their widow's peak off. If you shave it off, you still have widow's peak. Just like if you buy those contacts that change your eye color is really still whatever it was before you put those contacts on. So if we look at this, you ever saw that old TV show, The Munsters? Eddie Munster has widow's peak. So you either have widow's peak or a straight hairline. Widow's peak is dominant. So if you have widow's peak, you could have two capital W's, or you could have a capital W and a lowercase w. If you have a straight hairline, then you have two lowercase w's. Now, if you have widow's peak, either all your gametes have big W, if your genotype is big W, or half of your gametes are big W, half of them are little w. And again, let's make this a little bit smaller. Now, if you have a straight hairline, all your gametes, are just little w. So again, you should have either the entire first line written down, widow's peak, big W, or big W, little w, and then all big W are half big W, half little w. Or you should have the entire second line written down, straight hairline, little w, all little w. Are there any questions about how we're filling this out so far? Okay, let's go back and look at our picture. The next trait is whether you have attached earlobes. If you have attached earlobes, where your earlobes attach, they either kind of go straight across into the side of your head or they actually go down a little bit. If you have detached earlobes, you have a part of the earlobe that hangs down below the point of attachment. It's bigger on some people, smaller on others, but if you have a section that hangs down below the point of attachment, you have detached earlobes. Notice when you write out the phenotype, you have to write out the whole thing. And we always have to write out both possibilities. The only way we can figure out what your genotype is we could look at a family tree. For instance, if you have detached earlobes, but your mother had attached earlobes, that means your mother was little e. So we know that you had to inherit a little e from your mother. So in that case, we can predict what you look like. So if you have detached earlobes, either all your eggs or sperm are big E, or half of them are big E, half of them are little e. Oops. If your earlobes are attached, they're all little e. So again, you're not going to write all this down. You're just going to write down whatever applies to you. So you're not going to write down both detached and attached, just whatever you have. Okay, the next characteristic is whether you have a bent little finger or whether you have a straight little finger. If you have a bent little finger, you want to look at that last joint on the little finger. It's called a phalanx. You want to look at that phalanx and it bends in towards the other fingers. And the way you can tell this is you want to hold your hands up so that your fingers are next to each other. If you look at my little finger, it's basically straight. It's basically parallel to the other fingers. So I have a straight little finger. If it twists in, and it's got to twist in at this last joint, then it's bent. Now keep in mind, you also have to take into account if you played volleyball or football or softball or something, and you broke your finger at some point, then you probably want to look at the other hand. Oops. Notice a lot of times the letters start with one of the characteristics, F for fingers, if you want to think of it that way. So notice whenever you're homozygous, whether you're homozygous dominant or homozygous recessive, all the gametes that you make are the same. If you're heterozygous, you can make two types of gametes, and keep in mind that they're always made in equal numbers. So it's 50-50. If we were looking at a situation where you could make four types of gametes, it would be 25% for each of them. So the next one we're looking at is hitchhiker's thumb. And again, we're contrasting this with having a straight thumb. In this case, if you have hitchhiker's thumb, and again, you don't want to look just at the nail, you actually want to look at the joint. If you look at the joint, for my thumb is straight at the joint and then it bends outward at the nail. Everybody's thumb bends outward at the nail. But if you have hitchhiker's thumb, your thumb bends outward down at that joint, at the knuckle. So in this case, a straight thumb is dominant and hitchhiker's thumb is recessive. So if you have a straight thumb, you're either big H or big H, little h. If you have hitchhiker's thumb, you're little h, little h. If you have a straight thumb, depending on which genotype you have, either all your gametes are big H or half of them are big H, half of them are, little h. If you have hitchhiker's thumb, they're all little h. Now, the next characteristic is whether you have mid-digital hair. For mid-digital hair, you want to take one of your fingers, and you don't want to look at the part of the finger that's closest to your palm. And you don't want to look at the part of the finger that the nail is on. If you think about bending your finger, you want to look at the middle joint. And to find out if you have mid-digital hair, you're going to check all your fingers, well, except your thumb. Your thumb only has two, but you're going to check all your fingers. You're going to check along here, and you're going to look for even one hair. If you have one hair, you have something called mid-digital hair. If you don't have this, you have no mid-digital hair. Technically, if we say you have no mid-digital hair, we should check your toes too, because this affects the fingers and the toes. So by now you should be starting to see a pattern with the gametes. And that pattern continues here. So if you have the dominant characteristic, unless we know for sure what your genotype is, there's always two possibilities for the gametes you can make. You can either make all the dominant allele gametes, or you can make half and half. If you're homozygous recessive, there's always only one possibility. Next, we're going to look at thumb crossing. And for thumb crossing, take your hands and interlace your fingers so that the left finger is always over the right finger, or the right finger is always over the left finger. Now, don't just switch the thumb, switch all your fingers. For most people, there's one way that if you don't think about it and you just do it, there's one way that you interlace your fingers. If you try it the other way, usually it even feels a little bit uncomfortable. So whichever one feels right to you, whichever one you do naturally, if you don't think about it, the two phenotypes here, the two things we can observe are either the left thumb on top or the right thumb on top. Okay, the left thumb on top is dominant. So again, you should be starting to see a pattern here with the genotypes. Let's come back to red-green color blindness in a moment. Let's do iris pigment because that's similar, and red-green color blindness is a little bit differently. Okay, your iris pigment is just what color are your eyes. So you just write down brown eyes, green eyes, blue eyes, whatever color your eyes are. Now, if I could spell, eyes basically fall into two categories. There are brown eyes and there are blue eyes, and I should have written eyes here. Blue eyes include different shades of blue. They're not all the same blue, different shades of gray. Brown eyes include brown, green, anything except blue or gray. Now, brown eye color is dominant. So brown eye color is caused by big B, or big B, little b. Blue eyes or gray eyes are caused by two little b's. So again, for gametes, if your genotype is big B, all your gametes, all your eggs or sperm would be big B. Or if you're big B, half of them would be big B, half of them would be little b. And then finally, if you're have blue eyes, if you're little b, then all the gametes that you make are little b. Now, notice for all the characteristics we've talked about so far, we haven't really cared whether you're a male or a female. And it doesn't really make any difference. If we would look at whether your earlobes are attached or detached. Men aren't more likely to have attached earlobes or detached earlobes than women are. There's no difference between the genders. But if we look at red-green color blindness, there is a difference between the genders. And the reason there's a difference between the genders is because the gene for red-green color blindness is on the X chromosome. Well, remember, women are XX. They get 2 X chromosomes. Men are XY. They get an X chromosome and a Y. So for this one, we're going to have to expand our chart a little bit. There's really 4 different types of genotypes or phenotypes. Now, how can we tell your phenotype? This is part of a test on color blindness. And if you look at these circles, what you see are some little dots, and they're colored in different shades of red, orange, pink, green. And people who are colorblind can't see the numbers in these circles. Should be able to see distinct numbers in several of these circles, 4 of them. One of the circles, there aren't numbers in. You should be able to see a 12 here, and a 42 here, and a 71 here, and a 6 here, and here is just kind of a zigzag pattern, no particular number. Now, people who are colorblind, who can't distinguish, this is a particular type of colorblindness called red-green colorblind, don't see red and green, they see different shades of gray, essentially. So here, you could either be a female with normal color vision. Let's make this a little bit smaller so we don't end up taking up too much space. So that's one possibility. You could be red-green I can spell here. You could be a red-green colorblind female. You could be a male with normal color vision. Or you could be a red-green colorblind male. Now I'm just going to cut and paste here. So if you're a female and you have normal color vision, what genotypes could you have? Well, you could be X big B, X big B. And notice when we're talking about something on the X chromosome, we always write it this way, an X with a letter that's written above the line. This is called a superscript. And when we write this superscript, what we want to think of when we're working a problem with X chromosomes, or if we would have a gene on the Y chromosome, this X with the big B is a unit. They stick together. The B is actually located, the B is the allele for this gene, and the gene is located on the X chromosome. So a woman with normal color vision could be X big B, or she could be X big B, X little b. What about a female who's colorblind? Well, for a female who's colorblind, there's really only one possibility. This female has to be X little b, X little b. That's the only possibility. Now, for a male who's colorblind, or a male with normal color vision first, I guess is who we have. He's X big B, but then for a male, his other chromosome is a Y. So notice we know his genotype exactly. And the reason we know it is because a male only has one X chromosome. So whatever is on his X shows up. Unlike with a female, it's not going to be hidden. Now, if we have a male and he's colorblind, he's X little b Y. So notice here, there's actually 4 possibilities. Because if you think back to when we talked about Mendel the other day, this, in a sense, this is looking at 2 characteristics at the same time. It's looking at your color vision, and it's also looking at your gender. So 2 characteristics simultaneously. Now, what about gametes? So let's look at their gametes. Well, if a female is X big B, then all her egg cells will carry X big B. But if she's X big B, X little b, then half of her egg cells will get the X with the big B. Half of her egg cells will get the X with the little b. Now, what about a female who's X little b, X little b? Well, we look at her, all of her egg cells will be X with a little b. So notice sometimes a female can make 2 types of eggs. Sometimes she can only make 1 when we're talking about something on the X chromosome. Now, if we look at a male can always make 2 types of sperm because half of his sperm are going to get the X chromosome, half are going to get the Y. And an X chromosome is never the same as a Y. That's why boys and girls are different. So for a male, half of his sperm get the X with the big B if he has normal vision. And half of his sperm get the Y. Now, if we look at a male who's colorblind, it's very similar. A male who's colorblind, half of his sperm, whoops. Half of his sperm get the X with, this time, a little b. half of his sperm get the Y. So one thing to notice, men have an X chromosome, but they inherit their X from their mother. So if we're looking at a man who's colorblind, which parent did he inherit it from? His mom, because it's on the X chromosome. Also, if he can see colors, he also inherited that from his mother. Anything that's on the X chromosome, a man inherits from his mother, because he doesn't inherit an X from his father. He inherits a Y from his father. So are there any questions about how you would fill out this chart? Keep in mind that for each of these, there's, two options. Other than for this one, there's 4 options. You're only going to tell me what is true for you. So fill this chart out appropriately for you. Don't put all the information on it. If this is me, the person in green, and I can't roll my tongue, I would not write this part in yellow down. So you're only going to put the part that applies to you down. You have all the information, so you have to kind of look at yourself and decide, what fits you, and then that's what will be on the part that you turn in. Are there any questions about what you're going to do to finish up this assignment? Basically, you're only going to put down 1 line from each of these cells that I filled in, whatever applies to you. Now, we need to keep this in mind, and I'm going to switch screen sharing for a moment. And now keep in mind that assignment is due by the end of the day today, so before midnight. And next we're going to work on some Mendelian genetics problems. We're going to do some of these in class, and you will have to fill out the others. They're not due until Thursday, so you've got some time, but we're going to work on them now. And if you want to, you can, I'll stay logged into the class even after we're done working our samples. You can log back in if you have a question, or you can stay logged in if you have a question and ask some questions. So what we want to keep in mind is what we have up here at the beginning. Now, we can work genetics problems backwards, but typically we're going to start out with some information about the parents' phenotypes. What did the parents look like? So think about the chart that we just filled in. We started out with your characteristics, your phenotype. Do you have a straight thumb or a hitchhiker's thumb? Do you have blue eyes or brown eyes? From the phenotypes, we can figure out their genotypes. Sometimes we can't figure out the genotypes completely. We saw some examples of that. From the genotypes, we can figure out the gametes you can make. With the gametes, we can figure out the offspring's genotypes. We can look at 2 parents and we can see if the father can make sperm that have this gene in them and the mother can make eggs that have these alleles, what are the possible children they could produce? So we can figure out the offspring's genotypes, and then we can figure out the offspring's phenotypes. But it's like all problems, just like problems in a math class or anything else. We always want to think about what information we're given at the beginning and what information we're being asked to find. So here's some information on eye color in problem number 1. And it tells us if the allele big I results in brown eyes. So big I, notice that is a capital letter, so that means that this is a dominant allele. So brown eyes are dominant, and little I allele causes blue eyes. So little I, or blue eyes, is recessive. And then it says, you know, here's what we're actually going to do in the problem, but most problems will tell us some information about what's dominant and what's recessive. Write as much of the genotype as you can for people who show the following phenotypes. Okay, so we have a woman with brown eyes. Now notice in genetics problems, one of the things to keep in mind, unless it mentions the X chromosome, we'll see an example of this at one point, but unless we have a problem that mentions the X chromosome, we generally don't want to worry about gender. So a lot of problems will say it's a woman, it's a man, it's a bull, it's a cow, whatever. We're not really worried about that. Okay, so what's the genotype of a woman with brown eyes? Well, she could be big eye, or she could be big eye, little eye. Either one of those is possibility. Because all we know for sure is she has brown eyes. And since big eye is dominant, if she's big eye, little eye, she will have brown eyes. And you wouldn't be able to look at these two women and you wouldn't say the woman whose genotype is big eye, you wouldn't say that she's got browner eyes or anything like that. We wouldn't be able to tell the genotype from just looking at the phenotype. What about a man with brown eyes if his father had blue eyes? Well, okay, we know he's got a big eye. Anybody with brown eyes has a big eye. That's what gives you brown eyes. But his father had blue eyes. Well, think about what having blue eyes means. If you have blue eyes, your genotype is little I, little I. So if your genotype is little I, that means that you could only pass down a little I to your children. So this guy had to inherit a little I from his father. So the only genotype that's possible for him is big I, little I. Now notice we could also figure out from this, his mother must have brown eyes because he got the big I from her. So what about the father of the man in B? He could be little I, and in fact, that's the only thing he could be. If you have blue eyes, that's the only possibility for your genotype, little I. There is no other possibility. Now, what can we say about the mother of the man in B? She could be big I, little I, or she could be big I. We don't know for sure. We know that she passed down a big I to her son. But that doesn't tell us whether she was heterozygous, big I, little I, or whether she was homozygous, big I. We can't say for sure. A woman with brown eyes if one of her daughters has blue eyes. So can anybody tell me what this woman's genotype would have to be if she has brown eyes, but she has a daughter with blue eyes? Well, if we think about this, if we break it down, she has brown eyes, so we know she's got a big eye. She has a daughter with blue eyes. Well, her daughter with blue eyes had to inherit a little I from dad and a little I from mom. So mom must be big I, little I. So sometimes when we know somebody's phenotype, we can figure out exactly what their genotype is. Sometimes, there's two possibilities. So now let's think about making gametes. And let's start out by looking at how many gametes you can make. So the key is when we look at gametes, we need to know a genotype to figure out how many gametes they can make. So if somebody is homozygous, this person can only make one gamete. And if you think about it, all their gametes will be big I. Now that was for somebody who's homozygous dominant. If somebody's homozygous recessive, well, guess what? It's exactly the same. They can only make one gamete. The only difference is all their eggs or sperm carry little I instead of big I. Now, what about someone who's big I, little I, who's heterozygous? Half of their eggs or sperm will carry the big I. Half of their eggs or sperm will carry the little I. Now, notice in this case, we're only looking at one characteristic. What if we're looking at more than one characteristic? Well, if we're looking at more than one characteristic, the key thing that we have to remember is that when you form an egg or a sperm, when you form a gamete, you only pass down one of each type of allele to your offspring. So if we look at somebody whose genotype is big I, big T, they can still only make one type of gamete. All their gametes will be big I, all their gametes will be big I, big T. Now, what about somebody who's big I, big T, and this should say little t, that's a typo, sorry. What about this person? Well, this person can make two types of gametes. And their two gametes are going to be 1/2 big I, big T, and 1/2 big I, little t. Now, here's another person who can only make one type of gamete. Now, there is a mathematical way that you can figure out how many types of gametes somebody can make. So let's look at how we could figure this out mathematically. I'm not sure we need to do it here. So there are two different I's. And what we're going to do is we're going to count the number of different letters for each gamete, and we're going to multiply these together. So here we've got 2, because there's 2 different I's, a big I and a little I, times 1, because there's only one T. Or I should say there's only one kind of T. So 2 times 1, 2 possibilities. Notice the most possibilities we can have with 2 alleles, or 2 genes, I should say, is we could have 2 different I's, and we could have 2 different T's. So we could have 4 different combinations, 4 different gametes. So we can have 1, we can have 2, we can have 4. But notice there would never be 3 possibilities. Because if you think about the numbers that we're multiplying, all the numbers that we're multiplying in here are going to either be 1s or 2s. So we could get 1 if we multiply 2 1s together. We could get 2 if we multiply a 1 and a 2. Or we could get 4 if we multiply 2 2s. But if we're just multiplying 1 and 2, there's no way we can get a 3. Notice that since we're making gametes, if you think about chromosome numbers, they always have half as many alleles as the genotype of the parent. So if we start with 4 letters, our genotypes, if you think about our samples, are always going to have 2. And when we make a gamete, it's going to have 1 I and 1 T in this example. Why? Because if you make an egg with 2 I's, what characteristic is your child going to get for the T? You'd have to hope that your egg with 2 I's got lucky and met up with a sperm with 2 T's, or your kid would have either too many alleles for something or not enough. So here we have some examples of that. Now, notice the only thing that gets tricky is when we start looking at our X and our Y chromosomes. And what we have to think about, remember what I said, an X with a big B or an X with a little b is a unit. So the question is, we're looking to see basically whether things are different or the same. An X with a big B is the same as an X with a big B. An X with a big B is different than an X with a little b. So when we have X big B, X little b, there's 2 different gametes. When we have X big B, X big B, there's only one type of gamete that woman can make. All of her eggs will get an X with a big B. So most of the others are similar to what we just did. Now, number 3 says how many different kinds of gametes. Let's change this question to make it a little bit different. Let's say, what gametes can be formed by individuals with the following genotypes? So if we think about the examples that we said before, these are this individual, if their genotype is big F, if they are homozygous for having that bent little finger, then all their eggs or sperm would get big F. If we have somebody who's heterozygous for having hitchhiker's thumb, then half their eggs or sperm would get the big H. Half their eggs or sperm would get the little h. So for our woman down here that we were talking about, all her eggs would get the X with the little b. So now let's actually work a type of monohybrid cross. So let's work a sample. And while we're working these problems, we're going to use blue to represent the father and red to represent the mother. I don't have a good pink on my computer, so we're using eye color again. So in people, the allele big I results in brown eyes, and the allele little I causes blue eyes. What's the genotype of a heterozygous man? So he's heterozygous, so his genotype is big I, little I. What's the genotype of a heterozygous woman? Okay, so her genotype is also big I, little I. What types of sperm can the man make? Okay, half of his sperm carry a big I. Half of his sperm carry a little I. What type of eggs can the woman make? Well, she has the same genotype, so that means she's going to be able to make the same type of eggs. So I'm just going to copy and paste and change the color to red. So then it says make a Punnett square to show a cross between the man and the woman. So when you make a Punnett square, you could think of it like making a spreadsheet. And what we're going to do is to make a table. So I'm going to put a table in here. Let's see. I meant to make my table 3 by 3, but I screwed up, so. So here's how we're going to set up a Punnett square. Okay, we're going to put the eggs that the mother can make across the top. I said it's kind of like a spreadsheet. So if you've ever worked with a spreadsheet, you know that there's always numbers and letters that tell you which row and which column you're in. So this is our Punnett square. We're going to put the different types of eggs that the mother can make across the top. We're going to put the eggs that the sperm that the father can make across the rows. I like to keep my colors because that way I can keep track of everything. Now, how do we fill this out? Well, we take this big I from the mom and we're going to carry it down. You know, I said think of this like a spreadsheet. We're going to carry it down to all the cells underneath that big I. We're going to take this little I, we're going to carry it down to all the cells underneath the little I. Take the big I from dad, and we're going to carry it across to everything that's in the same row. We're going to take the big little I from dad and do the same thing. Now notice, if you don't have a program where you can make tables very easily, you can do the same thing just by using tabs. So I'm going to do this just using tabs. And basically the only difference is it doesn't have the little, I guess they call them borders for the cells. So we're going to do the same thing. We're going to carry the big I from the mom down underneath. We're going to carry the little I from the mom we're going to carry the big I from the dad across to every cell in the same row. And whoops, that should have been a little I there. And we're going to carry this. Now, if we look at these 4 spaces in here, or the 4 cells up here. These represent all the possibilities for our children. Notice that these cells are the same as these. So what do we predict for the genotypes of our offspring? So notice there's 4 possibilities for what our offspring get. Now up to this point, we've been keeping track of mom and dad, but now we're going to quit keeping track of mom and dad. I only see 1 out of the 4 possibilities that has 2 eyes. So we're going to say 1/4 of them that are 2 big eyes, sorry. 1/4 of them are big eye, big eye. I see 2 of them that have a capital I and a little I. So I'm going to say 2/4 are big eye, little I. And then I see 1 that's little I, little I. So 1/4 is little I, little I. Now notice this has to add up to 4/4, because if it adds up to 4/4, that means I counted everything in our Punnett square, but it also means I only counted things once. So we don't want to count things more than once. So if we add these up when we're done and they only add up to 3/4, we didn't count something. Or if they add up to 5/4, we counted something twice. Now notice, this is really just what your odds are of having a child with this genotype anytime a couple like this has a child. It doesn't necessarily mean this couple is going to have 4 children. They might have 1 child. They might not have any. They might have 10. And everybody knows a family where even though your odds are about 50-50 of having a boy or a girl, I have 3 brothers. I have no sisters. I know a woman who has 11 sisters and no brothers. So they had 12 girls in a row. So what this does mean, though, is overall, if we look at thousands or hundreds of thousands of births, yeah, about 1/4 of them ought to be big eye, big eye. About 2/4 of them ought to be big eye, little eye. But for one family, yeah, it doesn't mean much, except, like I say, this is your odds. This is why sometimes people make a mistake. They say, well, you know what? You know, you've had 4 boys in a row. You really want a girl. So if you get pregnant this time, it's got to be a girl. The odds are with you. Actually, the odds are about 50-50 again. And in fact, if you've had 4 boys in a row and you're trying to have a girl, probably your odds are slightly better than 50% of having a boy. There's probably something that's biasing the results in that direction. So what do you predict for the phenotypes of the offspring? Okay, so if we look at these 2 genotypes, big eye, and big eye, little I, remember, big eye is dominant. So we're going to predict that 3/4 of them will have brown eyes. So we're going to group those 2 together. Now the last category that we have is 1/4 of them are little I, little I. And we don't have anything to add that to. That's the only one that fits into that category. So 1/4 of them will be blue eyes. Now, there are several other questions with problems like this. These are all problems that are monohybrid crosses. Remember what we said the other day. Monohybrid crosses means we're looking at only one genetic characteristic. But now let's look at a dihybrid cross. So let's look at letter E as an example. Now let's look at D. That'll be a better one. So let's look at D. And notice that the question on D is, what would you predict for the genotype and phenotypes of the offspring? And anytime you see that, that basically means you're going to have to go through all the steps that we did here. Because notice in this sample, the last two problems are, what do you predict for the genotypes of the offspring? What do you predict for the phenotypes? So this means that we're going to have to do this. So let's look at the two guinea pigs that we're crossing here. And notice it tells us up at the top that black hair is big B, dominant. White hair is little b, recessive. Short hair is big F and big S, dominant. Long hair is little s, recessive. So it doesn't tell us which parent is which, so we'll just arbitrarily make the first parent the father and the second one the mother. So a guinea pig that is heterozygous for both characteristics. So the father is big B, big S, little s. So that's the father's genotype. Now, what's the mother's genotype? Well, the mother's genotype, it tells us that she has white hair. So up above, white hair is recessive, so she's little b, but she's heterozygous for hair length, so she's big S, little s. So let's be a little more specific here. Okay, so for the father's gametes, we can be more specific since it's a he, and we can say sperm. He's going to be able to make 4 types of gametes. A fourth of them will be big B with big S. A fourth of them will be big B with little s. A fourth of them will be little b with big S. And a fourth of them will be little b with little s. Now we can think about the mother's eggs. Well, notice she can only make 2 types of eggs. Remember, we said the way we figured out the number was we multiply the number of different B's times the number of different S's. So that's how we figured out 4 for the father. But she can only make little b's. So half of her eggs will get little b with big S. Half of her eggs will get little b with little s. Okay, so now we get to our Punnett square. So let's make a table here. And like I say, you could just use tabs. I find doing a table easier. So we said we're going to put the gametes for the mother, the eggs across the top. Notice that we still have to keep the 2 letters together, because if you think about the baby guinea pigs, they have to get an allele for hair color from mom, and they also have to get an allele for hair length from mom. So we're still going to fill out our Punnett square the same way we did before. We're going to carry the alleles from the mother down to every cell underneath them. Okay, now for the father, we have 4 possibilities we said. Oops, I wrote that one wrong. That should have been big B, little s. So again, we're going to carry the gametes from the father, the sperm, across to every row in the same row that they start in. Okay, this one's going to be a little bit trickier because we've got so many letters in there. So let's go through and start looking at what the offspring are going to look like. Okay, we've got 8 possible offspring. So that means our fractions are going to be 8ths. So an 1/8 of our offspring, and notice we've been writing our genotypes with B's first and then S's. So an 1/8 of our offspring have big B, little b, and then 2 little s's. And we have so many here. I'm gonna highlight them as we count them so that we don't count things twice. So let's see if we have anything else with with a big B, a little b, and 2 little s's. I don't think we do. That's the only one I see. So we may have to add something to it later. So I'm just going to kind of go across the cells like in order from top to bottom. So we have an 1/8 of them that are big B, little b, big S, little s. And actually, if I look down here, it looks like this one is also big B, big S, little s. So that means there's really 2/8 of these. Then I have big B, little b, and then 2 little s's. Okay, I don't think I see any more with that. Then I have 2 little b's, two big s's, then I have 2 little b's, big S, little s, and it looks like there are 2 of those. And then finally, I've got one down here. I don't think I need to highlight it. It's the last one. It's 2 little b's, 2 little s's. Okay, now we can go through and check our work at least in one way because there's 8 boxes, so our fractions are 8ths. So before, when we had 4 boxes, we said everything had to add up to 4/4. Now everything has to add up to 8/8. So 1, 3, 4, 5, 7, 8/8. So at least we know we didn't count anything twice or not count anything at all. So let's think about everything that has a big B and a big S in it. So if we look at these, ones, they have at least one big B, they have at least one big S. So this means when we look at these, there are 3/8 that have black short hair. Because big B gives them black hair, big S gives them short hair. Now there's 1/8 that have a big B, so they have black hair, but they have 2 little s's, so they have long hair. And notice when you work a problem, you don't have to use all the highlights like I am. I'm just doing it so we can keep track of what we have here. Then we have 3/8 that have 2 little b's, so they have white. Yes, and then they have short hair because they have at least one big S. And then finally we have 1/8 that have white. Long hair. So in a dihybrid cross, what we have to keep in mind, remember what we said the other day when we discussed Mendel, is we have to do 2 characteristics at once. We can't just do one. Are there any questions about how we worked some of these problems? Okay, the rest of these are going to be homework. They're due Thursday. And there's also a mitosis homework that's due tomorrow. So what you want to do is give me the homework that we worked on together. Hopefully you've got that filled in since we talked about it. And then I will repost this because on these problems, some of the problems now you have an answer for. So I'll repost this as soon as we're done. I won't repost it under assignments. I'm just going to repost it under content. But you can use this as the basis for your assignment if you want. And then you'll have a few of the questions already done. You'll have the samples done, and you'll have this dihybrid cross and a few samples on some of these earlier ones. And notice that we changed the question on number 3 to say what gametes can be formed. So I'll post this in just a moment, but before we do that, are there any questions? If there aren't any questions, I will go ahead and let you go for the day then.
Hello?
Hi.
Oh, I got a question.
Sure.
You talking about the chromosomes assignment?
I haven't graded it yet. I haven't seen it. Did you just send it in last night or what?
No, you said the one we worked on together. That's the one you were talking about, right?
Yeah, that was the one that I hadn't seen yet when we were talking.
Wait, no, I mean like we worked on it in class, didn't we? I think we did.
We might have done part of it in class. I'm not sure if we did all of it in class. I don't know. We might have done one problem. I don't remember.
You were talking about the one we just worked on together today then, right?
No. The chromosome assignment is something different. That's the chromosome numbers. That's probably back in last week. Let me check and see where it is.
Okay. Yeah, I was trying to contact the office yesterday to take a W, but I couldn't contact them. I probably was too late. I left a voicemail, but they're probably not going to do nothing. I think it's too late.
Well,
'Cause
your
yesterday
average is actually up around 64% now, so it's inching closer towards a C. I don't know if you'll.
I gotta put those homework assignments in.
Yeah, the chromosome assignment is actually in last week, in week 6.
Yeah,
So that's where you want to double check for it.
yeah. I don't know. I have problems like understanding this stuff. I might just like watch the videos again, but
Yeah, if that works for you, or I mean, double check too. I mean,
or
I don't
get
know
some
if you've
time with
looked.
you.
Yeah, if you check on YouTube, 2 of the ones that I really like are 2 girls called the Amoeba Sisters. Some of the explanations they give are a little bit simple, but in terms of just getting to understand it, I think that's good. And then there's another place called Khan Academy. Their lectures are a little more complicated. They're probably closer to what we do. But sometimes somebody else can explain something in a way that clicks for you a little bit better.
Okay.
So yeah, I mean, I'm not trying to discourage you from rewatching ours, but you might want to try watching some different ones too.
Okay. All right. I guess I'll try that.
Yeah, I think it's worth a shot.
All right.
I mean, if you look at 1 or 2 and they don't help you, then you can forget about it.
Okay. Because it's too late to withdraw now, so I have to do it anyway.
Yeah, I think you would have to get a grade now. I mean, you could check and see. I know they do make exceptions sometimes, but it's usually when people have some kind of medical issue or something like that, and they have a hard time getting out.
Okay.
I mean, I would say depending on how you do on the last 2 tests, the last test is probably actually the best test you've had so far. But depending how you do on the last 2 tests, I think for a while there, I felt like it was touch and go, but whether you're going to pass, but I think now you're definitely going to pass. So it's just a matter of, if you get a C or a D.
On the final, what do you mean that?
Yeah, for your final grade.
All right, Dan. Thank you.
You're welcome. Talk to you later.
All right, talk to you later.